Hello students, so as I mentioned to you guys last week, I was going to record these presentations, but since we haven't found any substitute teacher for this week, I'm going to record this anyways. So we are going to talk about blood in this presentation. Alright, so goals for this class. We are going to understand the basics of hematopoiesis, which means the creation of blood. What are the forms, elements in blood, the structure and function, origin, and fate of the red blood cells, including the breakdown of them, understanding the significance of hematocrit, and basic types of anemia. Describe the major components of plasma, including the major types of plasma proteins and their functions. We're gonna describe the function of the thrombocytes and platelet plugs during the hemostasis. Then we're gonna talk about the overall functions of the clotting cascade, which is very complicated, so we are not going to go into detail. Describe the basics of our normal clotting and the endogenous anticoagulants. Describe the basic types of blood and the RH factor. And then we should be able to recognize 7 form elements in the blood. And having all that taken care of, we should be able to understand what we're going to see in the lab. So what is blood? So blood is a connective tissue, because think about it. Don't think about something connective that is solid. This is something that is going to connect via transportation of form elements, as well as oxygen and other important gases and other nutrients throughout our body. It's sticky, it's viscous in consistency, and it has a metallic taste. And this is because of the iron that is present in the blood. So it's 5 times more viscous than water, hence the saying. And then its color is going to vary, so from bright red to deep dark red. And if you guys think about it, if it's bright red, it's rich in oxygen. If it's deep dark red, it's low on oxygen levels. So the blood itself is slightly alkaline, and I have talked to you guys about the physiological pH. So blood should be within this ranges from 7.35 to 7.45. This is called the physiological pH. Temperature, it's slightly warmer than outside of the body. So it's 1 degree Celsius more or warmer than the rest of the body. And accounts for 8% of total body weight. Amounts will vary among females and males. So in an adult male will be 5.6 liters, and in females will be 4.5 liters. So blood has important functions, as you guys know. It carries oxygen throughout our body from the lungs, known as external respiration, right? Then it carries nutrients from the GI tract. It carries carbon dioxide and other wastes to be removed from the cells throughout our body, and also carries endocrine hormones to their target tissues, meaning that if we have a endocrine functioning cell, is going to release some hormones and travel throughout the bloodstream to a target cell, and that's how it works. Then jumping from transportation to regulation helps regulate temperature and pH, so it stays at a homeostatic level, meaning everything has to be balanced, right? Also helps us to maintain fluid levels, and also has a protective function, meaning that it's going to prevent excessive blood loss by clotting. But also it's going to assist with immunity, right? So we are familiar with the white cells, but there are other form elements in our body that is going to help us with protection, meaning antibodies, which are familiar with. The complement proteins, think about this just as like pieces of proteins that are carried through our blood, and they target foreign elements so they can be recognized by the white cells and be destroyed, or the antibodies. And also we have the white cells, which are here, okay? So where does blood come from? Well, it all depends what stage of your life is. In utero, so you are not born yet. Not born yet, right? It's gonna be made in the liver and the spleen while we are not born, right? After birth, it's gonna be formed by the red bone marrow. Okay, so where is the red bone marrow? Usually it will be in the spongy parts of the long bones, and there's one that is gonna be doing that, and will be the, I'm just gonna put the first three letters, the femur. Okay, especially the spongy parts of the femur, and this is going to be known as red bone marrow. Okay, so approximately 100 billion new cells are made each day. Just think about the sheer quantity of cells being produced. So before differentiating, meaning that we're going to have a single cell that is going to differentiate in different kinds of cells, right? This is called differentiation. The blood cells, all blood cells are going to be hemopoietic stem cells. Okay? So it will be, as I said before, a cell. Think about like a blank slate that before the differentiation process is the blank slate that is going to turn into different cells, and this cell is going to be called hemopoietic stem cell. Okay, so keep on the hemopoietic stem cell. So let's just break this down. Hemo means blood, poiesis or poietic means production of something, and then we got the stem cells. So if you think about it, just breaking this term down is cells that produce blood, right? So this process, or these cells, I should say, these cells are also called hemocytoblast. We are familiar with, we are familiar with some of the terms here. So we have spoken about hemo, meaning blood, cyto, meaning cell, blast. Whenever you see blast at the end, or in some medical term, it's an immature cell. And this makes sense because this cell is not yet mature to differentiate, because it has to become something else at the end, right? So from this single cell, you guys get to see it turns into different stuff. All right? So these undifferentiated cells, known as stem cells, known as hemopoietic stem cells or hemocytoblasts, are going to have two different types of blood, meaning this first types of blood cells, right? So we're going to have a lymphoid cells, meaning these ones here, that are going to turn into the lymphoblast and then into the lymphocytes, right? And then we have the myeloid cells. This forms the other types of the blood cells. Some from the myeloid stem cell line, and you guys will hear that. We have a myeloid stem cell line and a lymphoid stem cell line. So from this line, we are going to have a myeloblast, and then this one is going to be turned into the granular white cells, as well as the platelets, as well as the red cells. Okay, so think about like the lymphoid stem cells line is going to produce the lymphocytes, and then the myeloid is going to produce the rest. Okay, so we are going to talk more into detail with the hematopoiesis in humans. As I said, hemato is blood, poiesis is production, right? So the production of blood in humans. So we have said that we have two main lines. So from the stem cell, which is a blank slate, think about an egg, that is going to be undifferentiated. At some point, a fertilized egg is going to differentiate and divide into specific cells that will create the baby chick, right? So, from this stem cell or hemocytoblast, we're going to have two main lines. So we're going to have the myeloid line and the lymphoid line. So let's talk first about the lymphoid line. So we're going to have a lymphoblast, which if it has still the blast part at the end, means that it's still not mature. From there, it's going to turn into a lymphocyte, and then it's going to be broken down into a natural killer, which is an immune cell that will attack foreign body defense, right? But also we have the small lymphocytes. Lymphocytes are gonna be differentiated into lymphocyte B and lymphocyte T. At this point, I have to talk where we are at in the process, right? So we have the bone marrow, blood, and then the tissue, right? All right. So the lymphocytes B, if they go to a certain part of the tissue, are going to be called plasma cells. And you guys will see that in some histological samples where there's a cell, very likely it could be a plasma cell. Okay? So what I want you guys to retain from all this big thing is the two main lines. What line produces the lymphocytes? What part produces the rest of the foreign elements in the blood? So just for last, there's kind of like a speed process where this lymphocyte cell is going to be turned into a lymphoid dendritic cells. All these guys have a common function, which is defense, right? And if you guys see the process here in the diagram, you'll see where they are at in the body, in the bone marrow, or in the blood, or in the tissue, right? Okay. So now we're just going to talk about the myeloid line. So if you see anything that says myeloid, means bone marrow. Okay? So, starting from this line, you're going to have the myeloblast, so still immature, and it's going to turn into megakaryocytes. You guys will see this very often in your textbooks. This is the final point. These are all form cells, meaning they're cells, right? But at this point, it's going to be broken down to thrombocytes. This guides, and I repeat, that they are not cells anymore. They are pieces of a cell, this guy. And this is what you see in blood. So it's not cell. Platelets are not cells. They're a wear part of a cell, okay? And of course, we know that those guys are going to be related to coagulation, meaning that they're necessary for blood clotting, right? Then, let's talk about this one. So, we have a proerythroblast. If it's blast, means that it's not mature yet, right? Then we have the erythroblast, and it's going to be changing from basophilic erythroblast to polychromatic erythroblast to orthochromatic erythroblasts, and then we're going to have polychromatic erythrocytes. At this point, the red cell, or the immature red cell, is not going to have a nucleus, right? And then they're going to turn into the red cell. As we have spoken before, red cells don't need nucleus or nuclei because they don't need it. That's not their function. Their function is to carry gases, right, and other elements. So they don't need a nucleus, okay? They lose it in this process. Then from this line, and this is something pretty important, we have mast cells, okay? Mast cells become mast cells in the tissue, and it's related to immunity, okay? So, coagulation, and think about this one as gas transportation, okay? Transportation, coagulation, okay? Coagulation, transportation. Think about it at those simple terms, okay? And this is the myeloid line, okay? So from this line, keeping still on the line, and the myeloid line, we're gonna have myeloblasts. They're gonna differentiate in 4 different types, okay? We are still in the bone marrow. I'm not gonna go into the detail of this, but if you guys think about it, we have a basophil, a neutrophil, and a eosinophil, okay? Then from this line, we're gonna get also monocytes. Monocytes are mature cells on themselves, but when these guys migrate to tissue, they turn into macrophages or myeloid dendritic cells. So it's similar to this one, but the thing that changes is their origin. They're either coming from the myeloid line or the lymphoid line. Okay? So you guys will see that a lot of these little cells here have little granules, and you will know these guys as granulocytes. Okay? And the ones that don't have those granules are called agranulocytes. Okay? All right, so what is blood made of? All right, so 55% of blood is plasma. Okay? The rest are the form elements of the blood. So we have in the form elements leukocytes and platelets, erythrocytes, which are the most dense components. So, form elements in the blood, cells and platelets, white cells, red cells, and platelets. Okay? Plasma, which is 55% of the whole blood, is going to be the least dense component, and this is where we're going to find the antibodies, a lot of protein, other elements that are going to be included in our immunity, but they are not cells. They're other structures. So think about the form elements of the blood as white cells and platelets and red cells, and then the rest in plasma. Okay? Okay, so components of blood, as we were talking, 2 major components, plasma and form elements. So let's go to plasma, which accounts for 55% of whole blood, meaning a lot of proteins and stuff that we don't need. And we need, let me correct that. We need that stuff, but it's not gonna be cells, okay? In the form elements, we're gonna have red cells, white cells, and platelets. The most common one of these form elements or cells is going to be the erythrocyte, okay? Then the leukocytes. Leukocytes are white cells. You will see that very often. Leuko, it's the medical term for white. erythro is red. So we have, let me just erase all this stuff. We know cyte means cell, right? So erythrocyte is red cell. Leukocyte is white cell. All right? So then we have the platelets, which are necessary for blood clotting, right? These guys are going to be located in the Buffy coat. Okay? Form elements, RBE, composing 45% of the blood. Plasma, 55% of the whole blood. So as we were talking before, whole blood comprehends 8% of our body weight. Breaking that whole blood, we have that 55% of our blood is blood plasma, and then 45% of that blood is form elements. Going into plasma, it's composed in its majority of water. So it's 91.5%. So we have to figure out the rest. Proteins compose 7% of plasma, and these proteins are going to be albumin, globulins, fibrinogen, and other stuff. So albumin is the richest by quantity and share of the plasma. Albumin helps us allow the transportation of certain hormones, as well as keeping balance of fluid within the blood vessels. I think about albumin, that's the way it sticks in my mind, is the same protein that you will see in eggs. So albumin eggs, that's how it sticks in my mind. And then we have globulins that helps with immunology and immune defense, because they carry certain structures that are going to help us with immune defense. Then fibrinogen, these guys are like little fibers of protein that's going to help us with the clotting functioning, and then other proteins. The other solutes, meaning what's left, are electrolytes, nutrients, gases, some regulatory substances, and waste products. Going to the 45%, we said that the red cells are the ones that have the major share of those form elements, right? Then we have white cells, and then we have platelets. Platelets are necessary for blood clotting, right? From the white cells, we have neutrophils, being 60 to 70% of our white cells. Then we have lymphocytes, 20 to 25%. Monocytes, 3 to 8%. Eosinophils, 2 to 4%. And then basophils, 0.5 to 1%. Okay, so here we are going to see a blood sample. So what they do is, with a drop of blood, they're going to stain it. And then, as we have seen on our histologic samples, they are going to react based on what it's in the cells. Anything that is purple are nuclei, right? Pinkish is the structures and contents in the cytoplasm. Okay? So, let's erase this. Here we are basically looking at form elements of the blood, right? Our most common ones and richest of them all are red cells, right? This clump here, at some point, were cells, but they're broken down. They're pieces of cells. Those are platelets, okay? Here, these big boys here are neutrophils, okay? And here, we have two types of lymphocytes, okay? We have lymphocyte here, and then we have a monocyte here. Okay? And this is how you will see it on a blood sample. Or they used to do this back in the day to do a blood count. Literally, they will go, grab a sample, and just start to count the cells. That's how they would do it back in the day. So here's more a fancier blood sample. This you can see with more detail what leukocytes look like. And of course they have different color because they are not stained, right? So leukocytes, then we have the erythrocytes, and then all those pieces that we were looking on the left of our previous slide are platelets, right? All right, blood plasma. We said it comprehends 55% of our whole blood, right? So mainly consists of water, above 90%. And then the plasma proteins comprehend from 7 to 8% of plasma, okay? The rest are electrolytes, meaning calcium, all the other cool ions such as phosphorus, potassium, all those other that we need for a lot of important stuff. Gases, meaning dissolved gases such as oxygen and carbon dioxide, nutrients and vitamins, and hormones. You guys remember in the endocrine function, that's how they're going to be transported or where they're going to be located while they get to their target spot, right? So plasma with plasma proteins removed is called serum. So if they don't have plasma proteins in it, it's called serum. Okay? So a little bit of a synopsis. Plasma comprehends 55% of our whole blood. Above 90% is water. 8% are proteins. The richest one is albumin. 2% or 1.5 to 2% of that plasma is electrolytes, gases, nutrients, vitamins, and hormones. Okay? Plasma without the plasma proteins is called serum. Okay? All right, so for blood plasma, this is the 8% that composes the plasma. So we're going to have albumin, globulins, and fibrinogen. So for the albumin, it's the most numerous one and the smallest one of plasma, composing around 60% of all plasma, right? So this one helps to regulate the osmotic pressure and also be a carrier for ions, hormones, lipids, and medication. Osmotic pressure relates to the functioning of keeping fluid within the vessels. The walls of the capillaries are very porous. So if there's not something to keep that fluid within the blood vessels, they're going to go to the tissues. And if they go to the tissues, we're going to have something very swollen and puffy. People that are malnourished have very low levels of protein, and that's why some of them, or a lot of them, look puffy in some areas, like the belly. Globulins, think about that as the antibodies. They're also the second most common one, protein. So they carry, and they have antibody proteins, but also the transport materials. And then the third most common one is the fibrinogen, which helps with blood clots. And the reason is like this fibrinogen, they're kind of like little fibers that are going to help us to create the blood plug. All right, so now let's talk about the form elements of blood. So form elements, as I said, think about the cells, are red cells, white cells, and thrombocytes, meaning the platelets, right? So two, as I told you guys, are cells because they're red cells and white cells. The third one, the thrombocytes, are platelets. They are no longer cells, they're pieces of cells. Okay? That's why they're not true cells. All right. So a lot of the cells are like final. They're so specialized that they lost their reproduction function or division function. So they have a short or determined lifespan. Okay. And like you guys know, these guys will die, these guys will die, and these are not cells anymore, so they don't reproduce. All right, so the erythrocytes are this amazing cells that are going to help us carry oxygen, and we know them for that. But also, they are shaped like a little, like, the rings, like lifesavers. They're flexible. Their size is 8 micrometers, and that's why they have the biconcave disc is that shape like lifesavers have. They have no nucleus because they lost it during that differentiation process because they don't need them, and they also have no organelles, like they don't have mitochondria or ribosomes because they lost that. Their function is different. Okay, that's why they can't reproduce, and they have a limited lifespan. So they're flexible, so they squeeze through small spaces, and they're just like that gooey, very flexible piece that goes to everywhere. They contain hemoglobin that is going to help us to carry those very important gases such as oxygen and bicarbonate dioxide, CO2. Okay? The hemoglobin is also be the responsible to give the red cell that beautiful red color. Okay? Hemoglobin. So hemoglobin contains 4 protein molecules called globins. Okay? It's a chain of 4. And they are containing of iron pigments. Okay? Attached to each protein. Think about this as 4 sets where oxygen molecules are going to go. Okay? So, the hemoglobin molecule can carry up to 4 molecules of oxygen. Okay? And as I said, they are 280 million hemoglobin molecules in one erythrocyte. That's, in my opinion, insane. All right? So, when the hemoglobin molecule is attached to oxygen, it's oxyhemoglobin. When it's attached to CO2 or without oxygen, it's deoxyhemoglobin. Okay? Without. All right. So, erythrocyte physiology. So, as we have spoken about, they are responsible to transport respiratory gases, O2 and CO2. Okay? A small amount of carbon dioxide is carried by hemoglobin, but it attaches to a different part of the hemoglobin protein, okay? It doesn't attach to the heme piece. All right. Erythropoiesis. This is the process where we are creating red cells. Poiesis, as I told you guys, is creation of something. Erythro is specific to the red cells, right? So these cells live 120 days because they lack of a nucleus and structures within a cell that a normal cell would just simply divide. Okay? So there's a hormone called erythropoietin that is going to help us to promote the production of red cells, meaning the erythropoiesis. Erythropoietin, and I'm going to erase this and leave just this bad boy here, is a hormone primarily located in the kidneys. Okay, liver to some amount, but kidney is going to be the main one to produce this hormone. Okay, and what it's going to do is those cells in the kidney are going to produce erythropoietin, and they're going to carry it and act upon think about this like the femur head. They're going to act upon the bone marrow to produce red cells, right? So then we're going to increase the production of red cells and have the whole line of production for red cells. What is affected by the red cells productions? Vitamin B12, which is absorbed by the intrinsic factor. Iron, our production of iron. Folic acid, because it's fundamental in the production of the hemoglobin and the red cells. Copper, because they also helps with the production of red cells. And vitamin C. Vitamin C is kind of like the cement for a lot of stuff. So a lot of athletes used to get doped with external erythropoietin because they will have more red cells, meaning if you have more red cells, the more you carry oxygen, the better your performance is. So these people were like performing better. Now that's banned. You will hear that kind of hormone as EPO, which is basically erythropoietin. All right, so here we're just seeing that erythropoiesis in humans. So the hormone gets to the bone marrow, to those stem cells, telling them, hey, I need to produce more red cells, and the process is going to start from the stem cell to the common myeloid progenitor, and then go through the whole process when the red cells are going to be produced and then released to the blood. Okay, and here's the process again. From stem cell to the whole process of the red cell. This is called differentiation for the cells. From a cell that has a nucleus and it's a stem cell, like a blank slate, going to like, let's say like red cell college. So you become a whole educated red cell. So this diagram is very helpful. So we're looking at basically hemostasis on the works. So hemostasis basically is a balance of any process. So it needs to have a stimulus that is going to work on the process until a balance is obtained, and so on. This never stops because things are constantly moving and there's always change, right? So this one in regards to the amount of red cells that are in our body, right? So the stimuli could be that I don't have enough oxygen in the air, so I need to make more red cells so I can obtain oxygen better. Maybe I lost enough red cells to carry that process, so I need more red cells. Maybe I don't have enough hemoglobin in those cells, so I need to produce more red cells so the hemoglobin carries oxygen better. Or there's decreased availability of oxygen, meaning like higher altitudes, so I need more red cells. So this stimuli is going to tell me, hey, cells in the kidney, I need erythropoietin. So that's produced, carried through the bloodstream to the bone marrow here, the red bone marrow. That is going to tell the stem cells, hey, I need to produce red cells, and the process begins. Those red cells are produced, differentiated, and then they are red cells in the system. Okay, so there's enough red cells to carry oxygen. Okay, maybe we don't need to produce as many because we have a balance. And then the whole process repeats when a stimuli is present to create the whole thing again. All right, so as we have talked about, red cells have a lifespan, 120 days. because as anything, they lose their flexibility, their hemoglobin degenerates, and they can't reproduce. So that means that they are gonna be unnecessary regardless, and we don't want anything that is not doing anything in our bodies to be just circling and doing nothing. So they start to degenerate and go to the spleen. They're broken down in the spleen, and then as anything that is being broken down is going to have some waste and some byproduct. So the iron in those molecules in the hemoglobin is going to be removed and stored in the liver and then returned to the blood. The remainder portion of the heme in the hemoglobin molecule is turned into bilirubin, and that one goes to additional changes, okay? And this is going to be either excreted, reused, or turned into something else, okay? The globulin portion of the hemoglobin is broken down into amino acids and also recycled in the body. So think about like the body is this very important and very miser system that is going to try to be as optimal as possible. Recycle stuff that we can reuse and then get rid of the other stuff that we don't need, okay? So 120 days, the red cells are not as youthful and full of energy as they were, and they're not as flexible, so we need to get rid of them, right? So it goes to the spleen where it's broken down, and those elements that are obtained for that breakdown of the red cells are going to be reused. Iron is going to be removed and stored in the liver to be returned to the blood. The remainder of the hemoglobin, or the heme portion of that hemoglobin, is going to turn into bilirubin, and you guys have heard that term before. And then that bilirubin is going to be changed into something else that is going to go to the GI tract and excreted in our feces, giving that brown color to our feces. And then the other globulin portion of the hemoglobin is going to be broken down into molecules such as amino acids and going to be recycled in the body for any other functioning. So here we're seeing what I just talked about, the hemoglobin breaking down. So the hemoglobin is going to be broken down into heme and globin, right? So the heme part is going to be broken down into iron that can be stored in the liver and then released to the blood. So this iron is going to be needed to the production of red cells. The bilirubin is going to be picked up by the liver, and guess what's going to happen with that? It's going to be turned into bile that is metabolized by the stercoraline, the bacteria, and is going to be excreted by the feces. This is what gives the poop the brown look that it has. Okay? Some of those metabolites from the bacteria processing of those erythrocytes are going to go back in the form of B12 and absorbed by the intestine, okay? And everything is reused again, okay? From the other side, so we have spoken about this whole process now, right? So the globin is going to be broken down into amino acids and is going to be carried in our blood. Not in the form of red cells, but in our plasma, and carried to other necessary stuff. All right, hematocrit. Hematocrit is basically the percentage of erythrocyte in our blood. Why is this necessary? Because for one, we need to make sure that we have enough amount of red cells in our blood to carry oxygen, right? But also we don't want too low, but we don't want too much. The more red cells are in our body, the more viscous our blood becomes. Meaning if I have a lot, I have a more viscous blood that is going to slow things down, and there's an increased risk of clotting and also plug in some areas. It's also going to vary among the genders. Males have more, 42% to 56%. Females are going to have 38 to 46%. Factors that involve or affect this are the amount of muscle, and that's why males have more. Also, things that affect the amount of red cells is the altitude. If you're in higher elevation, there's less oxygen available, which means we need to make more blood cells. So this is an adaptation that people up in the mountains have, so they can absorb of be more effective getting that oxygen. I grew up in the mountains and I have this adaptation, but once I moved to a higher elevation, I didn't need it anymore because the availability of oxygen was higher, so I didn't need much red cells, so I lost that availability. If you go to a higher elevation, you can adapt to that, making more blood cells, but it takes time. So anemia in our body means that the blood's oxygen carrying capacity is too low to support metabolism. Okay, so this is a big thing. So there are three main groups of anemia, and they're not listed here, but they are microcytic, meaning small cells, hypochromic, or microcytic, meaning that the macrocytic are cells that are too big, and hypochromic, that they don't have enough of the hemoglobin function to carry oxygen. And this is going to be caused by different stuff. If I don't have enough blood cells because I'm losing them, it's a cause of anemia. There's anemia where I don't have enough red cells. There is one where our red cells are being destroyed too fast, meaning maybe some immune response, or maybe the structure of our red cells is not effective enough. Inadequate production of red cells, not enough iron, or not enough red cells. These guys are going to affect and cause one of these three major types of anemia: microcytic, hyperchromic, or macrocytic. Okay? Symptoms: yellow eyes, because there's a lot of bilirubin going through our blood system in the destruction of cells. I'm feeling fatigued, dizzy, because I don't have enough oxygen being carried. There's not enough blood, so I have low blood pressure. The heart is working faster because they need to pump more blood. Maybe there's pain, maybe there's a heart attack. The spleen is working and overdriving because maybe a lot of those red cells are being destroyed and it's being stored here. Intestinal reasons: I'm weak. I don't have enough oxygen to just function. Shortness of breath because there is not enough oxygen to be carried, and I need oxygen to move around. If there's not enough blood, in oxygen, I'm gonna look pale. I'm gonna be cold. I'm gonna be a little bit yellow because maybe a little bit of destruction of those red cells. So now we have the different problem. We have polycythemia, right? A lot of cells in the blood. So increased amount of erythrocytes in our blood is going to make our blood more viscous. And this can be due to dehydration because there's not enough water, so it gets concentrated. Smoking and air pollution is going to alter the production of red cells. High altitude, as I told you guys about, is the adaptation, where the body tries to be more effective in the carrying and catching that oxygen since it's less available. And then excessive exercise, because if we're increasing our demand for oxygen because of intense activity, we need more red cells. People used to get doped in the meaning like getting maybe more erythropoietin to produce more red cells, and that will increase their performance. A guy who was very famous for doing this was Lance Armstrong, who was doping with erythropoietin to produce more red cells and be better at those bike races. So the sickle cell disease is basically when our red cells don't have this shape, but they have this shape instead, like a half moon. Of course, we cannot work with this because we need the flexibility and the form of the cells to go through capillaries and other crevices, right? And here, this is what we see here. So in a normal physiological speaking way, red cells will go this way without any problem. With the sickle cell disease, what happens is like you still have the normal ones, but you have the presence of this kinds of cells in our blood system. So they have the tendency to clog and clump in some places. And what's going to happen is that it can lead to heart attacks, joint pain, swelling of hands and feet, bacterial infections, dizziness, and even stroke. Okay? People with sickle cell disease will have something called a sickle cell crisis, which causes a lot of pain. Okay? The reason that people experience this or develop this condition is because at some point people adapted, genetically speaking, to develop these kinds of cells because malaria cannot deposit here as easy as this one. It was a great adaptation, but with the unintended consequence of this. All right, so now let's turn our attention to leukocytes. Leukocytes are white cells. Leuko means white, cyte means cells, right? So these guys have been differentiated, but they have nuclei and organelle. They're bigger also than the red cells, and their function is to initiate response and also defend against our bodies, against pathogens, right? So they use the blood as transportation, and white cells just live a few days on average, so they have a shorter lifespan than the red cells, right? So there are two kinds of white cells or leukocytes. You will have your granular leukocytes and agranular leukocytes. They have granules or they don't. Okay? In the granular group, we have three different kinds. As you guys can see in these pictures, they have granules, right? These granules have different functions. Some of them contain some chemicals that can kill the pathogen. Some of them have chemicals that can induce inflammation. Some of them have peroxide that can kill some pathogens. So there is a reason why they are there. Okay, so the main three types are neutrophils, basophils, and eosinophils. Okay, so we have here the neutrophil, we got the eosinophil, and the basophil. Okay, the further slides are gonna go into detail to each kind. Okay, so the first one is the neutrophil. This one is the most common one of the leukocytes. Okay, the most abundant of the leukocytes, meaning the most abundant about white cells. Okay, they are active during acute systemic infections. Okay, so just short time, very sudden. Okay, they control, remember this, bacterial infections and engulf bacteria, so they literally will like eat them. Literally, they're bigger than red cells, and you will spot a neutrophil very easy because they have this 3 to 6 lobes and they will be connected. Okay, then you have eosinophils, bigger than the red cells, and when I looked at them, usually they have like 2 lobes, and your books will say they has like the shape of a telephone receiver. For me, they look like a Spider-Man. All right, they're also a little bit more red. They're reddish, or redder than the other ones, and they are active against parasitic worm infections. Okay, keep that in mind. Against parasitic worm infections. Okay, they also play a role in allergies and asthma. Okay. All right, so the third kind is the basophil and is the least abundant of these guys, okay? So they look very particularly because this is the cell and it has a nucleus and then the rest of the cytoplasm, right? But there's so many granules in this guy that you cannot tell one from the other. So the reason that these granules are there because they're very rich in two main components, histamine and heparin, and they are related to inflammatory response and the allergic reaction. So when there's an allergic reaction, these guys are going to be very rich and present there. When you have an allergy, like very chronic allergy, you take antihistaminics, right? You want to inhibit this. Okay? All right, so then we have our three boys together back again. So we have the neutrophil, the most common one. And you guys will see that has like the chain nuclei together, all right? And you'll see it here, right? This is what it is. And then we have the eosinophil here. It has like that Spider-Man kind of like nuclei, if you guys think about it. It's this one here that you will see here. And it's bigger than the other ones. Then you have your basophil, which you will see more granules more than anything, and that's how you can spot it because it's super rich in those granules. And you'll see it here too, okay? This one is the least common, so if you see it, lucky. But also, they should not be as abundant in normal red cells because, again, they're present in allergic reactions. They are the least common of the white cells. Okay? So with the agranular leukocytes are the cells, the white cells that don't have that obvious sand-looking granules in their cytoplasm, right? Like the case of the basophil that is super rich in those sand-like granules, or the neutrophils when it has like all those itty-bitty spots, right? So there are two main kinds. We have lymphocytes and monocytes, okay? The first one is the lymphocyte. It's the second most common white cell. It's large and purple. And you can tell from like, apart from like the neutrophil because it has a huge nuclei, okay? And there are three main kinds. So you have the T cell, the B cell, and natural killer, okay? So lymphocytes, especially the T cells, attack virus-infected cells, okay, and tumor cells. The B cells come from plasma and create antibodies, okay? And then the natural killers are that like innate immunity and they attack tissue. They are very important also to regulate the possibility of that some tumor cells grow and then it becomes cancer, right? Okay, perfect. So T cells, B cells, and natural killers, they're all lymphocytes. Monocytes are the other ones, and you will see it because they're massive. They're chunkers, okay? And they have the phagocytic function becoming the macrophages, and we have seen an example of this bad boy on the lining of the alveoli, okay? So they become active during chronic infections opposed as acute infections, okay? All right, and here we see a photo of them. So you have your monocyte, chunky, big, and then we have the lymphocyte. See how big this guy is, but also the nuclei is, but there's not as many sandy looking as the granulocytes. This bad boy here, you can see it's more nucleus more than anything, and I can tell that's a lymphocyte. Okay, so how common is each white cell? So I told you guys before, a way I remember it is one I know from all the white cells. I know the neutrophils are the most common ones, but how I remember the rest, I say N L M E B. Now let's make eggs and bacon. Let me repeat that silly way that I remember this. Now, because it's neutrophils, let's, for lymphocytes, make from monocytes, eggs, or E for eosinophils, B, bacon for basophils. That way, I remember what's first. So, neutrophils, lymphocytes, monocytes, and basophils. So, neutrophils make 50 to 70% lymphocytes, 20 to 35% monocytes, 4 to 8% eosinophils, 2 to 4%, and then basophils, 1%. Okay? Neutrophils, lymphocytes, monocytes, basophils. Eosinophils and basophils. Okay? Here we have the gang together. So we have neutrophils, that you guys see it here. Then we have the eosinophil, and as I keep saying, it's redder, but also has kind of like a Spider-Man looking shape. And then we have the basophil, so if you guys can see, it's full of granules, so you cannot see the nucleus. Then we have the lymphocytes here, and it has a big old nuclei, and that's why you can tell that part. And then you have the monocyte, which is big, and it also has a huge nuclei, okay? So leukocytosis, let's break this term down. Tosis means there's a condition. Cyto means that we're talking about cells, right? Leuko, white cells. So there is something going on with white cells, okay? The total count of white cells should be no greater than 11,000, okay? So if there's above 11,000, there's something going on, and it could be due to different reasons. Infection, because we need the white cells to fight that infection. An allergic reaction, because something triggered that allergic reaction, meaning an allergen. So the cells that have that histamine are fighting against it. Or conditions caused by dehydration or increased stress. Imagine that. Leukopenia, anything that is penia, we're lacking of, or we have low amounts. So not enough white cells, right? So the total count is below normal, okay? And that can be due to different reasons. Metal poisoning, radiation, or sickness. Our bodies are either radiated to the point that the white cells are not being produced enough, or we are sick long enough that those reserves are depleted. Measles affect the production of white cells. Mumps, same thing. Chickenpox, yes. AIDS. The function of the AIDS virus of HIV is to deplete the white cells. And if you don't have white cells, you become very, very, very vulnerable to infections. Chemotherapy is going to deplete everything that is growing, including the white cells. So as I said, the leukopenia is going to put the patient at higher risk to contract infections because we don't have enough of the white cells to defend our bodies. Leukemia. White cells in our blood. But this one is the condition when there's a overproduction of abnormal white cells. Okay? So these cells are not doing what they're supposed to do. They don't look that they're supposed to do. And there are different types. And of course, they can be either mortal or not if they're treated. 90% of all leukemias are diagnosed in adults. Symptoms of leukemia: anemia, bleeding, fever, weight loss, fatigue, and bone pain, because we are working those bones, especially the femur, to produce white cells. Survival rates can be from only like 90% to 100% in months, but it will depend then of the type of leukemia. Okay, it all depends when it's caught, what kind of leukemia is, and how it's treated. All right, so we have the thrombocytes. They're fragments of the megakaryocytes, and they help us with the clotting process. Okay, and also to repair damaged blood vessels. Okay, the hormone that controls the thrombocyte production is the thrombopoietin. Okay, and they're produced primarily in the liver and kidneys. Okay, they're a quarter of the size of a red cell, and that's why they're itty bitty, because they're only chunks of a cell. Okay, here we have a table, and this one has a description of all the form elements in the blood, from the red cells to the white cells to the thrombocytes. So I suggest you guys take a detailed look at this table. All right, so hemostasis. Hemo means blood. Stasis is mean that it's getting still in one spot, right? So this means that is the process that we need to have in order to stop bleeding. And this can be because the injury of a blood vessel, meaning a cut. This means that it's going to help us to prevent hemorrhage, which is the large amount of blood loss. Okay, so when large blood vessels are damaged, of course, medical intervention is going to be required, because the hemostasis is not going to make it, friends. So the process consists of 3 major steps: vascular spasm, the plaque formation, and then coagulation. Okay, and we're going to talk about it in the next. So let's say a little vessel gets damaged or cut. So what's going to happen, as we were talking of any other vessels, meaning arterioles, veins, or arteries, they all have muscle layers, because we need to cause something called vasoconstriction. In this case, the vasoconstriction is going to be caused because there's an injury of that vessel. That is going to trigger the response of contraction, a vasoconstriction. Also, the endothelium, meaning the internal lining of that vessel, is going to be exposed. So those collagen fibers are going to make more sticky to the platelets to adhere. The platelets are going to release chemicals. They're going to make other nearby platelets stickier. And what's going to happen is that we're going to have a plug forming here, right? Then the fibrin proteins are going to make a mesh. So we're talking about those fibrinogen proteins just like floating around our plasma. They're going to stick and form a mesh that is going to trap some red cells, but also platelets, and create a clot. Having this clot created is going to help us to do what? A clot. Helping us not letting blood out. Okay? Okay, so we continue talking about the hemostasis. Here is just more like we talked about in the diagram, but in text. So the vascular spasm, or the vasoconstriction, means that these damaged vessels are going to contract. And then that also helps to reduce the blood loss. Then the collagen exposed in that endothelium is going to attract platelets. The platelets are going to release some chemicals that are going to tell the platelets, hey, we need some help here. And they're going to adhere to those platelets, creating a platelet plug. Then with the fibrinogen that we're going to talk in the next slide, So as we were talking, the platelet plug is going to be created, but needs to be reinforced by fibrin. But in order to get fibrin, there's a few things that we need to have. Okay. So first, we're going to have prothrombin activator being created, and it can be within the blood or by external factors such as a medication. Okay. So then, the prothrombin is going to be turned into thrombin, but it needs the presence of this activator, which is the guy that we just spoke about here. Okay? And we can see it here. So prothrombin is going to be turned into thrombin. The thrombin is going to react with the fibrinogen or fibrinogen that is in our plasma to create fibrin. Okay? And this is also aided by certain factors. Okay? So now we have a platelet plugged that is going to be aided by fibrin to create a clot. And this is necessary because we don't want this whole thing happening while we don't need it, because we're going to clot everywhere. And of course, we don't want that. Okay? So the platelet plug, it's going to create all this. So the crisis was taken care of by the clot, right? But of course, we got to get rid of all the unnecessary clots once the heal process has happened. And this is where the enzyme plasmin goes into place. So this is a natural clot buster, and it takes usually 2 days after the injury. Okay? And basically what it does is, like, see this mesh here? This is going to be released, destroyed. That way there are not clots flowing everywhere, because we don't want clots in our blood. And we're going to talk about that later. So clotting disorders. So we have talked about those clots that are formed and they're not completely destroyed. If a thrombus is still unbroken in our blood vessels, this is going to be a problem, because the thrombus can turn to an embolus and then go to our bloodstream. And what's going to happen is that the embolus is going to clog some artery when it's small enough to occlude the supply of blood. And that's dangerous, because it fits a little artery that is working in our heart or our brain, is going to cause an embolism, which is going to result in the dying of that area, which of course we know it's dangerous because it can be mortal. Hence, pulmonary embolism, or the blockage of an artery in the heart or an artery in the brain, which can be mortal in those cases. Other blood clotting disorders. Let's say that there's a thrombus because we've been laying in bed all day in the hospital and we have not been doing anything to prevent that. It's going to create a thrombus in those veins in our vessels, right? So that thrombus is going to get loose and go to our lungs, where it can be mortal. So what we do is, 1, prevent it by moving. 2, compression stockings. 3, by giving enoxaparin, so this never happens. Okay? Maybe we have the opposite problem. Maybe we're not clotting. This is when we have hemophilia, which is an inherited disease. Back in the day, people used to die from this all the time, because if your father have something, you were not stopping bleeding, creating the whole clot cascade, so you will bleed to death. Okay, so now we are going to talk about the blood types. As we were talking previously in our class, we have antigens or antibodies. Antigens are markers. Think about it as a little badge in the wall cell that is gonna be like an identifier for the antibodies to attach. Okay, so in the blood, the antigens are called agglutinogens, and in the blood, those antibodies are gonna be given the name of agglutinins. Okay, continuing in the blood typing. So if the red cells have type A antigens, they're known as type A. And if they have antigens, meaning those receptors in their cells, it's known as type B. If they have both of these guys, we know it as AB blood, right? But what if you don't have any? You're O, nothing, zero. So no antigens. So let's go again. You have antigens type A, you're type A. If you have antigens type B, you're type B. If you have both AB, you're AB. And if you don't have anything, you're O. A little more of a visual. So those little dots on the cell walls are the antigens, which are receptors. So type A, you have the antigens type A. If you have type B antigens, you're type B. If you have both, you're AB. And if you don't have anything, you are O. Okay, so this is when it gets a little bit complicated, right? So we have type A, as we have established, you have receptors, A, but you have antibodies against B. If you have type A blood, you have antibodies for A. If you have type AB blood, you have no antibodies for neither of these guys. Because if you had them, you will be attacking your own blood. If you don't have the receptors, meaning that you are type O, you have both antibodies. Because you don't have the receptors for this, but you have the antibodies for all those other guys. Let's see an image in the next slide. Okay, so you have the receptors A, antigen A. You have the antibodies for B. You have the antigen, the receptors for B. You have the antibodies for A. But if you have both antigens, meaning A and B, you don't want any antibodies because you will be destroying your own blood. The opposite is for type O. You don't have any, but you want to have the antibodies for the other blood types. Okay? All right. So this is another layer. On top of the antigens that you also have an Rh type antigen, also known as the antigen. Okay? And this is the Rh type. And it's named after this little cute monkey here, the monkey Rhesus. Okay? If you do have the antigen, you're positive. Okay? Meaning if you're type A and you have the antigen, you are type A positive. If you have B and you also have the antigen, you're A positive. But on the other hand, if you're type B but don't have the antigen, you're B negative. But if you have A and no antigen, you're A negative. When it comes down to this, is also for blood transfusion. If you have the receptor, it's kind of the same situation with AB. You don't want antibodies for this because you don't want to destroy your own blood, right? So you can receive blood from both types, right? But if you don't have the receptor, meaning the antigen, you are going to have antibodies for that blood that is B positive, right? So you cannot receive blood from this guy, because what's going to happen is, since you are having those antibodies, it's going to attack that blood and it's going to be destroyed. And you're going to die. So let's repeat this. If you have the receptor, meaning the Rh receptor, you're A positive, okay? If you don't have it, you're A negative. If you are A negative, you are going to have the antibody for the Rh factor, right? Meaning that if you get any, let's say, any positive blood, meaning if you're A negative and you get A positive, that's a no-no, because you are going to attack that person's blood. So meaning that if you're A or B negative, you can only receive Rh negative blood. But if you're A positive or B positive or AB positive, you can receive your equivalent, either positive or negative type. This is just the percentages. I think it's a little bit dated, but this is just for your information, okay? All right, blood testing. So type AB. You can see here that there is agglutination for both, okay? Type A, you can see it here. Type B, you see it here. Type O doesn't have it because there are no receptors for those antibodies to attach. Okay? Okay, back in the day, blood transfusions were a coin flip, because we didn't have blood typing before, right? So blood transfusion is what it looks like. It's transferring blood from one person into another person. So if you receive blood that is not compatible with yours, you're going to have agglutination, which is going to be the clumping of the blood, which is going to damage your brain, kidneys, and around the heart. If you are O negative, meaning that you don't have neither receptors for A or B or Rh, you are universal donor. If you have A, B positive blood, you're a universal acceptor, because you have the receptors for B, A and B, but also you have receptors for positive. So you don't have any antibodies for any blood that is given to you. Let's repeat that. If you are O negative, meaning your blood, the red cells you are giving away, don't have any receptors for anything. So you're basically a blank slate, right? You are a universal donor. If you are AB and positive, your blood has receptors for A, B, and the Rh factor. You don't have any antibodies for type A, type B, or type or a Rh positive, meaning that you can receive any blood. Okay? Okay, so this is a little like table about what we just talked about. All right, if you're type A, your antigens, of course, are going to be A. You have antibodies for B. you are compatible to receive blood with A and O. If you have blood type B, you have, of course, antigens for B, but you have antibodies for A. And you can receive blood from B and O. Okay? Of course, you cannot receive blood from either A and B because you have the antibodies for both. Okay? Same goes with these guys here. If you have the antibodies for B, you cannot receive B blood, but that goes because if you have AB, it's going to attack the receptors for B. Okay? All right? If you have AB, your antigens are A and B. You don't have antibodies for neither A or B. And that's the beauty of being a universal receptor, because you can receive A, B, AB, and O. Okay? And you're the universal receptor, so you're cool. You're cool. And then for the type O, you don't have receptors for neither A or B. You have antibodies for A and B, and you're compatible with only O. You're incompatible with the rest. Okay? All right, another diagram. So if you're O, you're universal donor, right? You can give to all these guys, right? If you are AB, you can receive from all of them because you don't have antibodies for neither of these guys, okay? So universal receptor, AB, O, negative, universal donor. Okay, so the hemolytic disease of the newborn, a dad that is positive has a baby with a mom that is negative. After the firstborn, if mom's immune system gets exposed to this Rh positive kid, immune response is going to create antibodies. When mom becomes pregnant for the second time with another Rh positive kid, this is going to cause an immune response when they're born, because the immune system is going to attack this Rh positive blood cells of this kid and is going to create a destruction of those red cells, which is going to be coming in anemia in the kid, increase the bilirubin, and possible brain damage or even death. What do we do? And kids used to die from this like back in the day, a lot. What we do nowadays for this? Well, we can give mom RhoGAM so mom doesn't develop the antibodies for this kid. So the next pregnancies are secure. Okay? Here we see a little diagram. So we got the positive kid, Rh positive kid from Rh negative mom, right? And dad is Rh positive. He's the responsible to give us this Rh positive baby. Baby's born. The antibodies are present. The next pregnancy, it's going to be the problem if we don't get the RhoGAM, right? And that concludes our blood presentation. Thank you.